Hello everyone, my name is Rahul. In the previous video, we had discussed about genetically engineered insulin. In this video, we are going to discuss about gene therapy. Gene therapy refers to a collection of methods. It refers to a collection of methods that allow correction of a genetic defect that has been diagnosed in a child or an embryo. So what normally happens is, for every enzyme that is produced in the body, we will have two genes, one from the paternal side and one from the maternal side. But there will be certain instances when there will be a single copy of the gene in case that gene is an X-linked or a Y-linked gene. So suppose that both the copies of the gene that have been inherited, they are mutated, or if there is a single copy in case of X or Y-linked gene that is mutated, then the particular function of that particular gene will be lost. So every gene, it will produce some enzyme or a protein. If this gene is mutated, it will lead to loss of function, which will lead to enzymatic defect. So we are going to study gene therapy with the help of an example of ADA deficiency. ADA stands for adenosine deaminase. It stands for adenosine deaminase. This enzyme, adenosine deaminase, is responsible for maturation and proper development of immune cells. For maturation and proper development of immune cells. If this enzyme is deficient, it will lead to a condition known as severe combined immunodeficiency disorder, or SCIDS, severe combined immunodeficiency disorder, or SCIDS. In this particular condition, all the immune cells will be absent and the person will suffer from frequent infections, which will be life-threatening since there will be no immunity against these infections. So the gene for ADA production is mutated in case of SCIDS. Now what we can do is, this ADA, it is deficient. What we can do is, we have something called as enzyme replacement therapy, in which functional enzyme will be injected into the patient and the condition will be cured. It will not be cured, it will be treated, because this enzyme, it will replace the faulty enzyme. Next option we have bone marrow transplantation. Bone marrow transplantation. Since this enzyme is being produced by bone marrow, we can replace the bone marrow by transplantation and our problem will be solved. However, the problem with both these approaches is that they are not completely curative. These approaches are not completely curative. Why? Because in enzyme replacement we will have to replace the enzyme again and again, and in bone marrow transplantation the results are not very satisfying. Only in a small proportion of cases we actually succeed, and in a large proportion of cases bone marrow transplantation will not cure the problem completely. It will only solve our problem partially. So the better option we have is gene therapy. We have is gene therapy. In gene therapy, what we do is we take lymphocytes from the patient. From the patient's blood, we will take some lymphocytes, and these lymphocytes, they are grown in a culture outside the body. These lymphocytes are then infected by a retroviral vector containing complementary DNA for ADA gene. This particular retrovirus we will produce beforehand. This will have the ADA producing gene, and we will have the virus infect these lymphocytes. So these lymphocytes now have the ability to produce ADA in lymphocytes. These lymphocytes are transferred to the patient. So the gene for ADA production is now present inside the patient's body. This gene will continue producing the ADA, or the adenosine deaminase enzyme, and when these lymphocytes will multiply inside the body, those lymphocytes, the progeny, they will also have this gene, and they will produce the required enzyme. So in a manner our problem is solved. We can cure the patient in this manner. However, there is a possibility that these lymphocytes are not immortal. They will fight the infection and they will invariably die. Since these lymphocytes are not immortal, we will have to periodically infuse the patient. We will have to periodically infuse the patient with these recombinant DNA containing lymphocytes. These lymphocytes are not immortal, so periodic infusion of lymphocytes is necessary. Now again, gene therapy seems to be not able to solve this problem completely. However, however, if gene which is isolated from the bone marrow cells is transferred to the embryo in early stages. So we are talking about isolating this gene from another patient, another person, where this is functioning well, or a synthetically synthesized gene. If this gene is transferred to embryo during the early embryonic stages, this can permanently cure the patient. Because we know that the embryo contains totipotent or the pluripotent cells. So if we have once put the gene inside the embryo during the early embryonic stages, this will cure the patient permanently. All the bone marrow cells will have this gene, and the patient will not require this periodic reinfusion of lymphocytes. However, experiments on embryo, they are currently banned we cannot produce the embryo like this. We cannot infuse genes into embryo. It is against the bioethics. It is not ethical to manipulate the human embryos. So what we prefer is this. This method of gene therapy is preferred, and the first clinical gene therapy was given in the year 1990 to a 4-year-old girl with ADA deficiency. This was the first instance of a gene therapy that was given. Now, in some previous years, in the last few years, there was a few Chinese scientists which claimed that they have produced human embryos resistant to HIV. They succeeded in providing resistance against HIV to the human embryo by the gene therapy, but their experiments were widely disregarded, and they are now suffering trials in their country. So it is against the ethics, it is against the laws of nature to manipulate the human embryo, because if we manipulate the embryo, we are essentially interfering with nature's work, and it could lead to disastrous and catastrophic results. So gene therapy in embryo is banned. In this manner, by providing gene to the lymphocytes, we can treat the ADA deficiency patients. So with this, we finish off with this topic on gene therapy. See you in next video.